Recall that diploid cells have pairs of homologous chromosomes. Those are two of each chromosome, one from the mother and one from the father. Now, meiosis has two rounds of cell division, the first being marked by the separation of the homologous chromosomes. So remember that meiosis 1 is the separation of these homologous chromosomes. However, each of these chromosomes still consists of the two sister chromatids joined by a centromere. Thus, the second round of cell division, meiosis 2, is the separation of those sister chromatids, resulting in 4 haploid gametes, just one of each chromosome in each cell. So meiosis, since it goes through 2 rounds of cell division, then we need to distinguish meiosis 1 from meiosis 2. And since each round of cell division is marked by the prophase, metaphase, anaphase, and telophase phases of division, then they must be numbered as well. So in prophase 1, we see a number of similarities to mitosis. There's the fact that the chromosomes condense into that chromatid form. The nucleus we see is dissolving, it has to go away, and the spindle is forming. Those are all similarities to mitosis. However, what's different from mitosis is the fact that these sister chromatids are called dyads, and what they do is they synapse with each other, the homologous chromosomes do, to form a tetrad. So two dyads find each other in the nucleus to synapse, calling that a tetrad. Now, what's the significance of this process? The significance is that because they get very close to each other, each of the pairs of homologous chromosomes, then what we have is a crossing over event, or an exchange of genetic material between these two chromosomes. So we see here at the tips in a couple of places that we've actually had a switch of some of the genetic material with each of these chromosomes. And this introduces genetic variation, which is very important. So now we move on to metaphase 1. And again, similar to mitosis, we see a number of things. First of all, there's a lining up at the spindle equator that occurs during metaphase. The nucleus is now gone. The spindle fibers have fully formed and are attached to the chromosomes at their centromeres. However, what's different from mitosis is that instead of just lining up each individual chromosome down the spindle equator, it's the pairs of homologous chromosomes that align at the spindle equator. They found each other in the nucleus, and now these pairs lined up beside each other are what are at the spindle equator. And of course, this is in preparation for their separation. And thus anaphase 1 is marked by the pulling apart of those homologous chromosomes. Again, so remember that meiosis 1, the whole point here is to get those homologous chromosomes apart from each other. And this really happens during anaphase 1. Now, this is different from mitosis because it's the homologous chromosomes that are separating. Mitosis, we just lined all the chromosomes up individually and separated their sister chromatids. Now we see that it's the pairs of chromosomes that pull apart. Now, in anaphase 1, we also see a very important phase that introduces genetic variation called independent assortment. So this is our second example. An independent assortment is the fact that these maternal and paternal chromosomes of a homologous pair move to one side of the cell or the other randomly. So what ends up happening is a very unique genetic combination, a mixture of chromosomes from the mother and the father end up getting enclosed in their own cells after meiosis 1. So if you look at these larger chromosomes, this homologue and this homologue, you can see that the one from the mom went to the left and the one from the dad went to the right. Opposite situation for the smaller one. However, those bigger chromosomes could have easily gone the opposite way than this. And now we see that what will be a future cell here has a very different set of genes than what we just saw in the former representation. And that is independent assortment. Okay, so then telophase 1, again very similar to mitosis in the way of these chromosomes are going to loosen into that thin chromatin form. The nuclei are going to form around each of the bundles of separated chromosomes as a result of anaphase 1, and the spindle is almost completely gone, as well as we see the beginning of cytokinesis, or the division of the cytoplasm. However, the big difference from mitosis here is that these nuclei are haploid because each of the homologous chromosomes have been separated from each other, and therefore there's only one set of chromosomes in each nucleus. The sister chromatids, remember, are still joined together, and pulling them apart is the point of meiosis II. So after a brief intermediate period called interkinesis, the entire process starts over again. And prophase II and metaphase II are pretty much the same events as mitosis, only with half the chromosome number. So again, we see the chromosomes condense, the nuclei disappear, and the chromosomes line up down the middle of the spindle equator. So in anaphase II, then, we get that separation of sister chromatids. And we pull apart the sister chromatids again, just like mitosis, to either side of the cell, except now we're following both of the daughter cells from meiosis I at the same time here. And then, as in telophase of mitosis, we will create new nuclei around each of these sides of the cell, encapsulating the new bundles of chromosomes, and the cell will complete division. And this will create, in the end, 4 haploid gametes produced by meiosis. And they're haploid because they have only one of each chromosome. Remember, we pulled apart those homologous chromosomes during meiosis I, and thus officially created haploid cells. And then with crossing over and independent assortment acting to increase genetic variation, we can also see how none of these 4 gametes that were produced at the end are similar to each other. None of them have the same combination of genes, even within just these 2 chromosome examples out of the total 23.